Hello everyone. In the second part of the seeds presentation, we will be going through the experiments and a few other terminologies. We have seen the conditions that were required for germination of seeds. In this experiment that was conducted, this cotton wool was dry. The cotton wool placed here was dry. Okay, and the seeds were put along with it. In this, the second one, the cotton wool that was placed here was wet, it was soaked in water, and then the seeds were placed here. So, we wanted to prove that water is required for germination of seeds. Without water, germination of seeds is not possible. So, when there was dry cotton wool, it was easy to understand and see that the seeds did not germinate. However, in case of wet cotton wool, after a little time, we could see that the seeds did germinate. This proves that water is required for germination of seeds. Apart from the cotton wool, when here it was dry, here it is wet, all the conditions that were kept were constant for A as well as B, both. Similar seeds, the similar temperature, all other conditions were kept constant. Except here, there was dry cotton wool and there was no germination. And here there was wet cotton wool, which did result in germination. This proves that water is necessary for germination of seeds. In the second experiment, here we want to prove that you need an optimal temperature, a suitable temperature for germination of seeds. If here there is wet cotton wool and the seeds are placed at an optimum temperature, then germination of seeds will be seen. However, if here the seeds are placed in an ice box in which the temperature is very low, then germination of seeds will not be seen. So here we can see that at an optimum temperature, at a suitable temperature, germination of seeds will be seen. However, at very low temperatures, germination of seeds will not be seen. It is to be understood that whenever you talk about germination of seeds, very low or very high temperatures create a lot of problems. So you only need an optimum temperature for the germination of seeds. If very low temperature is present, like in this ice box, then it does not allow the embryo to grow. And if very high temperature is there, then also it would not allow the germination of seeds as the tissues would be destructed. So for germination of seeds, only optimum temperature is good. In this third experiment, we wanted to prove that oxygen is required for germination. We wanted to prove that oxygen is required for germination. In this case, in A, we have placed alkaline pyrogallic acid. This is alkaline pyrogallic acid. It absorbs oxygen. It absorbs oxygen. You have moist cotton wool here, okay, and you have the seeds. Here you only have water, but here you have alkaline pyrogallic acid. Now, because this alkaline pyrogallic acid has absorbed all the oxygen, no germination will be seen in the seeds of A. However, in case of B, there is water and there is presence of oxygen. So, it is possible to see the germination of seeds. So in B, the seeds would germinate. However, in A, because of alkaline pyrogallic acid absorbing the oxygen, germination would not be possible. This proves that oxygen is required for germination of seeds. This experiment is called as the three beans experiment. I repeat, this experiment is called as the three beans experiment. This seed is completely inside water. The seed is completely inside water. This seed is halfway in water, halfway in air. And this seed, the third one, is completely in air. Dip the seed underwater. This halfway in water, halfway in air. And this seed is completely in air. These seeds are attached to a glass slide and they are placed here. Now, if you leave this in a warm place, you will see that the middle seed is the one that is germinating. The middle seed germinates because it gets oxygen from the air and it gets water. It's halfway in water, halfway in air, right? So it gets oxygen as well as water. But when you talk about the top seed, that is the one that is only present in air, it has complete access to oxygen but not to water. Complete access to oxygen but not to water. For this reason, it will not germinate. The seed that is inside the water would probably germinate, but the germination would be very, very limited. This is because it is getting water and some form of oxygen from it. Water as well as some amount of oxygen from it. However, the oxygen supply would eventually run out and germination would not be very significant. So therefore, the significant germination is seen in a seed that is halfway in water and halfway in air. So basically, whenever you talk about germination of seeds, seed itself contains a dormant embryo. When you talk about a dormant embryo, it is an embryo that is inactive. It is an embryo that is inactive. After it undergoes maturation, after it undergoes maturation, this embryo would form a seedling. This embryo would form a seedling. So basically, the embryo is dormant, and then on becoming active, it would form a seedling. When you talk about germination, there are two types of germination. You have the hypogeal germination and you have the epigeal germination. Always remember the epicotyle and the hypocotyle of a seed will never elongate together. Either the epicotyle elongates or the hypocotyle elongates. So, if the epicotyle elongates, the cotyledons will remain under the ground. The cotyledons will remain under the ground. And you would call this the hypogeal germination. If the epicotyle elongates, then the germination is called as hypogeal germination and the cotyledons remain underground. If the hypocotyle elongates, the cotyledons are pushed above the ground. If the hypocotyle elongates, the cotyledons are pushed above the ground, and this is called as the epigeal germination. So when you talk about the hypogeal germination, the cotyledons remain underground, and the epicotyle is the one that is elongating faster. Remember one thing: the hypogeal germination is usually seen in monocotyledonous seeds, and the epigeal germination, in which the cotyledons are pushed above the ground as the hypocotyle gets faster, is seen mostly in dicotyledonous seeds. Hypogeal germination usually in monocotyledonous seeds, and epigeal germination is mostly seen in dicotyledonous seeds. We next come to viviparous germination. This is a very special form of germination. You can see it in plants like Rhizophora and Soneraceae. Now, in this special type of germination, okay, which is viviparous germination, the seed will germinate while it is inside the fruit. I repeat, the germination of seed is seen while it is inside the fruit and when it is still attached to the parent plant. The attachment is still there to the parent plant, and inside the fruit, the seed germinates. Post germination, okay, the plant drops the seedling into the soil. Now, the seedling fixes itself into the soil and develops the roots. It is seen in plants like Rhizophora and Soneraceae, wherein the seed germinates inside the fruit, okay, while it is still attached to the parent plant. The seedling is then dropped into the soil, where it fixes itself and develops the fruit. When you use the term seedling, it is a stage in the growth of the plant from a seed before it has become wholly independent of the food stored in it. Right before, right before it becomes completely independent of the food stored in it, is seedling. It is seedling. The roots of the seedling do start absorbing water and minerals. The roots of a seedling do start absorbing water and minerals. Okay, and the leaves would then start manufacturing food through the process called as photosynthesis. The roots absorb water and minerals, and the leaves would start producing food through the process of photosynthesis. Okay, after growing, it will become a mature plant and produce its own flowers and later seeds. Thank you.